Welcome back, everyone. It's my pleasure to introduce our third speaker of the morning, and this is Professor Hans Klever. Hans is the Director of Research at the Princess Maxima Center for Pediatric Oncology. He clearly is one of our speakers where it's impossible for me to even summarize his accomplishments. He's won over a dozen major international scientific awards recognizing his pioneering work. He is a prolific investigator, I think approaching almost 400 publications. And just this year, I don't get a chance to read that much anymore, but I have personally had a chance to read 2 of your beautiful reviews and an original research article, which I hope you will talk more about today, Stem Cell-Grown Organoids as Models for Human Disease. Hans, welcome.
Well, thanks very much for a brief but kind introduction, and thanks also for the invitation. I've been a member of the Gairdner jury in the past years. Actually been here I believe in January. I've never been during the major gala event yesterday, so I was very pleasantly surprised by the event, but also by the temperature, because the 3 times I was here was something like -10 walking from the hotel to Anyway, so I'll be talking about the gut. We already heard from Emma. This is a small intestine. Colon would be very similar, but it has a flat surface. This is called a villus. It's a surface extension that helps us to digest food very efficiently and take up nutrients. And when we first saw this structure, we realized it was probably the best structure in a vertebrate body, in a mammalian body, to study the activity of adult stem cells. And there are several reasons. First of all, the speed. Cells are born near the base of the crypts down there. They proliferate for 2 days, so there was presumed to be a stem cell at the base of the crypts. They proliferate for 2 days, go through 5 or 6 cell cycles, then leave the crypts, differentiate out very rapidly into one of 6 or 7 cell types. You see them here. And by the age of 4 days, these cells then reach the tips of the villi and they undergo apoptosis. So it's an extremely rapidly cell-renewing tissue. There is a massive amount of epithelium in your gut, so you produce about 100 grams of tissue every day. So the speed was interesting to us, but also the very stereotypical design of crypts, so about 10 crypts surrounding a single villus, and the fact that this unit is repeated about 1 million times in a mouse gut and about 1 billion times in the gut of a human being. So we thought this is really the best system, by the way it's designed and by its speed, to try to understand exactly what the rules are that control normal stem cell biology. We had an entry, I don't show this here, but totally by chance we discovered, we cloned the transcription factors that are the effectors of the Wnt pathway. We showed that when you knock out one of them, TCF4, these mice die at birth and they have no crypts. So that was probably the first evidence that Wnt maintain adult stem cells in crypts. We now know that Wnt maintain the stem cells, we believe, of all epithelia in our body, ectodermal, mesodermal, endodermal. And at the same time, in a collaboration with Vogelstein, we found that colon cancer comes about by the inappropriate activation of the same Wnt signaling pathway. And then by very carefully comparing gene expression profiles between cancers and normal crypts, and this was done in a collaboration with Pat Brown, we actually found a marker, LGR5, it's in this slide, that then defined, this is much, much later, helped us define the stem cells and then create mice that showed us exactly what the rules are that control these stem cells. Now, LGR5 encodes, this is all very recent, it encodes a 7-transmembrane receptor. These are the Wnt receptors, and it turns out that, first of all, LGR5 is a fantastic marker. We believe, again, for all active epithelial cells in our body, but it also performs an important function. It is a receptor for R-spondin, and R-spondin are secreted molecules first defined by Christoph Niers in Germany as potent amplifiers of Wnt signals. There was no real biochemical understanding of how they would do this, and this is where LGR5 and R-spondin come in. And also 2 very recently identified E3 ligases, seen by Feng Kong in Boston and by my lab. And to actually summarize a lot of blots and non-interesting biochemistry, this is the story. So here you see the classical Wnt receptors, frizzled and LRP. They will bind secreted Wnt's. Wnt's are lipid modified, are very difficult to work with, very hard to produce. They don't travel very, very far in tissues. When you engage their receptors through beta-catenin and TCF, they activate target genes. And amongst those target genes are these 2 E3 ligases that actually will then very specifically ubiquitinate the frizzles and remove them from the surface. Now, this is a very, very rapid negative feedback loop. In about 1 or 2 hours, a wind signal is off. And this is fantastic during development, where cells very rapidly need to switch fates. But adult stem cells need much more. They need a robust, prolonged wind signal. This is where LGR5 comes in, and our spondin. So our spondin with its second furan domain binds LGR5. With its first furan domain will bind to these E3 ligases and then breaks this negative feedback loop. So when both are present, so when you're a stem cell and you have LGR5 and there is our spondin around, you now keep your activated Wnt receptors on the surface. And if you read this out by TCF reporters that we designed a long time ago, the signal goes up from about 5-fold to 500-fold. So it's an incredibly potent enhancer of Wnt signals. On their own, I stress, our spondins and LGR5s, as far as we can tell, do nothing. You really need a pre-existing Wnt signal and then they act. This is all very recent. It explains a lot of the experiments I'll show you, but actually we were just very lucky to do them the way we did them. Now, Nick Barker made a number of knock-in mice, and I don't show you all the details, but I hope you can see here on the left 3 or 4 villi, 5 or 6 scripts. This is a mouse where we can we inserted GFP into the LGR5 locus, and this revealed the presence of these tiny cells at the very base of the crypt that we and almost everybody in the field had entirely missed. We also have QER in there. We can do lineage tracing for the insiders. And essentially, to summarize a lot of papers on these cells, they didn't fit any of the characteristics that stem cell biologists had sort of come up with based on the hemobiotic system. So they're not quiescent, first of all. That's very important. They divide every day. Logistics of this is enormous. We believe that our gut stem cells divide 20,000 consecutive times, and they do so apparently without undergoing too many mutations, because not all of us will get colon cancer in our 80 or 90 years of life on Earth. But we don't exactly know how they do this. So they're not quiescent. This is a sort of a defining characteristic for many stem cell biologists. They're not rare. Up to 10% of crypt cells are these cells. And actually they're all identical and they all are potential stem cells. You do not divide asymmetrically. You'll find in your textbooks that's what stem cells see. Well, but they do actually, if you read the literature, there are really no good examples of mammalian stem cells where people have really visualized this or studied this. The stem cell hierarchy is not unidirectional. Again, I don't show this, but often many cells in a stem cell hierarchy can move back and forth in the hierarchy, and they can actually, when you lose your stem cells, they can go all the way back and become a stem cell. And I'll give you an example of this. Again, the classical thinking that a stem cell is rare, non-dividing, and unique, so if you lose it, it's the end of that particular tissue. No, that's not the case. In many, many tissues, you can actually kill the stem cells, and other cell types, even fully differentiated cells, sometimes can go all the way back to become stem cells. And then in the gut, and probably in other tissues, we know that the system itself often creates the niche, and in our case, Paneth cells, which are daughters of the stem cells, are also the cells that provide many of the growth factors that the stem cells need. Now, we can sort from these mice. We can sort these LGR5 positive cells at the base of the crypt. So this is one of these crypts. You see here the green cells, purified stem cells here, all done by Nick Barker in the lab, who is now in Singapore. Toshi Sato then, based on this very unexpected observation that these cells are constantly dividing, decided to see if he could do something that what is regularly done by ES cells or IPS cells, which is essentially take them in culture, give them a limited set of defined growth factors, and then continuously grow them as stem cells. Now, the dogma was that adult stem cells don't do this, yet Howard Green, for instance, at Harvard in the 80s already showed that epidermal stem cells, although he was not allowed to use that term, epidermal stem cells can expand massively and still stay normal. So we thought that we would actually starting from a single cell, we would make large numbers of stem cells, but we got something very different. We start first creating these rather chaotic structures. We only need 3 proteins here, so our spondin, we picked it because it was a Wnt agonist. We did not know it was the ligand of our LGR5 receptor at that time. We knew that EGF was important. We knew from an old experiment that we needed to block BMP signaling, and this is sort of a generic culture system that allows you to grow almost all epithelia. They can usually be improved a lot by adding or changing stuff, but this is the major component, Wnt, tyrosine kinase receptor ligand, inhibition of TGF-beta BMP, and we do this in a gel, Matrigel. Matthias Lutolf has shown you can actually replace Matrigel by a fully defined hydrogel that carries specific peptides. And under these conditions, we now have these structures growing for 7 years. They don't look like balls of stem cells. They have a central lumen, as you can see here. There's multiple buds that stick out, and by using various microscopic techniques and single-cell sequencing, we essentially find that these stem cells, so a single stem cell recreates a mini version of the gut. I should stress it's only the epithelium, it's not the muscle so it's only epithelial cells that we can grow, but all the cell types are there, and by single-cell sequencing, Alexander van Oudenaarde actually found 2 cell types that were not known, secreting sort of hormones that were not known to be expressed in the gut. And we then went back from the mini gut to a real gut. We found that they existed in real life. So it's really it is a complete phenocopy of the structures that exist in a living mouse. Stem cells, planet cells at the base of crypts, rapid transiently amplifying daughter cells, and even the life cycle of being born here, proliferate for 2 days, differentiate out, live for another 2 days, and end in the lumen of the gut is retained between the mini guts and the real guts. Now, based on these observations, Toshi and many other people in the lab came up with protocols, and we're still in the process of publishing all of these for essentially, we believe now, all human epithelia. And again, we need to activate Wnt, we need to activate the tyrosine kinase receptor, depending on what tissue it is. We almost always need to block TGF-beta and BMP. Sometimes it's good to have BMP activity in the culture medium. We add steroids like estrogens or progesterone or testosterone, depending on if it's prostate or it's breast or ovary. But there's a whole list now of protocols. Other labs have added more. Taste bud was not done by us, but these taste buds actually taste. You can inject sweet or salt, and they start signaling through calcium and cyclic AMP. And as far as we can tell, they are genetically entirely normal. Telomeres stay long, mutation rate is really low, transplant done, and liver has been done back in. So human they integrate in the normal tissue, and they never, what would have been predicted, will cause a cancer or will be exhausted and disappear. So this is all very, very surprising. We don't know exactly how these stem cells do this. We're working hard on it, but they know the tissue that they were derived from. We'll give you a few examples. In the stomach, this is an example. Again, that doesn't fit at all the classical hemopoietic system of stem cell hierarchies. These structures are not called crypts, they're called glands in the epithelium of the stomach. They contain the cells that lower the pH, the parietal cells, to about 1 or 2, the cells that produce pepsinogen, called the chief cells. There's goblet cells, there's hormone-secreting cells. There is a region that's called the isthmus where proliferation takes place. The renewal is not as fast as it's in the gut. It's in the order in a mouse of maybe 4 or 5 weeks. Some cell types live much longer. Lots of arguments over who knows what the stem cell is here. The isthmus cells, there are too many of them according to classical definition, so they cannot be the stem cell. They're always proliferative. Yet we believe they maintain the healthy tissue. But on top of that, there is the chief cell. Some of you might have heard of this cell before. It makes pepsinogen. It's a beautiful cell, fully mature. It actually had been seen by Leblanc in Quebec to be proliferative at a very, very slow rate. They divide once every 3 or 4 months. But when we perform a lineage tracing experiment, we can mark using LTR5 or TROY, a similar marker. We can mark one of these cells, and then we see that at a very slow rate. And if you wait for about a year, you'll see this happening. The entire gland is replaced by cells that come from this fully differentiated cell. It's at the end of the hierarchy. Now, this is quantitatively very unimportant, given the amounts of cells in the healthy state. However, if you lose your isthmus cells, which would be after chemotherapy or radiation, this kickstarts the process of the chief cells to repair the tissue, and in a matter of weeks now these cells will replace the entire system. So these chief cells, make pepsinogen, that's their function, but they moonlight as reserve stem cells and are called into action when I think many other examples now in kidney and in lung and in liver where similar processes take place. And I stuck this in because we've been experimenting to see if we can culture microbes inside these mini structures, following on Emma's story this morning. So here we grow from one human chief cell, we grow human stomach organoids. And, the slide is not here, but we can now inject Helicobacter, and Helicobacter will functionally infect these organoids, colonize these organoids, drill through this thick mucus layer, and engage the cells the way they normally engage cells in the stomach of an infected individual. Other labs, Maria Estes has shown you can use these things to grow norovirus. Could never be cultured on cell lines, but works fantastically well on human organoids grown in this procedure. And we, for instance, recently got Cryptosporidium to complete its life cycle. Cryptosporidium is a malaria-related protozoan that nobody culture very well, particularly sexual cycles. If you inject a few oocysts in stomach or colon organoids, they will actually complete their entire life cycle and for long times produce Cryptosporidium infectious particles. Another example, the airways, what you see here is in green the cilia. So every patch is a cell that has the cilia, and as you know, the airways of the lungs produce mucus, and then the cilia brush up the mucus into your oral cavity. That's how you keep your lungs dust-free. We can grow human lung organoids from sputum, so we don't even need a biopsy. We just spin down some live cells, and then we get these organoids. We were first surprised to see that they have mucus, as you can see here, but that the mucus was moving. It normally doesn't happen in colon or small intestine or stomach. And then when they blow up, we realize that they have functional cilia, and these cilia somehow connect, and they beat. They produce these waves, productive waves of beating. And when you actually push them together, they'll make tubes. They'll fuse quite easily, and then all the cilia line up in the longitudinal axis of the tube. So, all very surprising. In the long run, in the very long run, we think that these types of cultured stem cells could be used, like people have been exploring iPS cells, and we heard of this earlier today, for regenerative medicine. We know they're genetically very stable. We know they're extremely well fated. So if you get them from the liver, they are really liver cells, they're nothing else, and they really resemble, even after a year, exactly what you would find in a fresh bit of tissue. In the short run, we actually hope to use this technology, or we're doing this, and I'll give you an example, for diagnostic purposes. Now we like one or two slides, and one of them is cystic fibrosis. So cystic fibrosis is the most common hereditary disease amongst Caucasians. It always affects the CFTR channel. In that sense, it's a very simple disease, always the same gene, yet about half the patients share the same mutation, so that would be one form of disease. There are mutations that are less common, but still would affect 3 to 5 to 8% of patients, depending on the country you live in. But then there's also a very large number of mutations that are rare, only occurring in individuals or families or villages or small ethnic groups. Vertex developed drugs, you probably have followed this, for the major mutations, rather remarkable drugs. So this is a group of mutations that affect the gating of the channel. It's a chloride channel, it doesn't open and close well, and a single small molecule for some of these mutations will actually correct the gating. So the molecule engages a receptor and then corrects the gating properties of that channel. And this large group is a group with a mutation that it lacks phenylalanine at position 508, is a misfolding protein. One small molecule, lumacaftor, will actually, as a chaperone, help the correct folding of the protein. The channel then gets to the surface, and then the other, ivacaftor, will support the normal gating of this channel. And for some, very unpredictably, for some of these patients, this works very well. Now, there's a number of problems with this. First of all, the price. Vertex charged in the US $250,000 per patient per year for this drug here, and was planning to charge double for the combination of two drugs, so $500,000 per patient per year. For this group, this small group, the trials indicate that it works for most of the individuals. For this much larger group, the trials indicate that only part of the patients really benefit. Some very well, some are a little bit helped, and many do not really respond at all. And clinically, you can only decide if it works by following a patient for a year, because they usually lose lung function by 3% every year, and you have to stop that you block the loss of lung function. So to show that you no longer lose 3% of lung function per year, which is a very, very complicated thing. Particularly, these are usually kids, they have lung infections, it's not easy to measure in these kids, etc., etc. So we came up with, and this is done in collaboration with the pediatricians in Utrecht, with an assay, very simple. We can do it on lung organoids, we can also do it on rectal organoids, which always become available in the process of a positive DNA test for young babies. These are healthy controls. One other thing I should say is that we believe, or it is believed, that cystic fibrosis is so common because it, being a carrier, is presumed to protect against cholera infections. And particularly in medieval times in Europe, there was a lot of cholera. The cholera toxin secreted by the bacterium opens the channel, this particular channel, and then actually you produce a massive diarrhea, and you might lose 20 liters of fluid every day, and then by dehydration, particularly the weaker individuals will rapidly die. And if you're heterozygous for a mutation in the channel, you don't produce this massive amount of diarrhea, and you have a higher chance to live, much like sickle cell anemia and malaria. Now, this is what you see here. On the left, we open the channel with cholera toxin or with forskolin, and very rapidly chloride ions get replaced, liquid follows, and you see the accumulation of liquid inside these organoids. That takes about 1 to 2 hours. We have software now that scans this and produces graphs, and is extremely reproducible between individuals and also from organoid to organoid. If you do the same test with a typical delta 508 homozygote CF patient, the organoids grow well, but you can already see they are collapsed. They have very little lumen. Probably the maintenance of a normal lumen already involves CFTR. And if we now add cholera toxin, nothing happens. If we now, for this particular individual, incubate the organoids for a few hours in the Vertex drugs, this particular here, we restore the response, and as a matter, it takes about 6 hours. And actually, we now have done about 300 or 400 patients, and every time that where we have clinical data, where we see this response, we know that clinically the patient will respond as well. And the test takes about a week, so we get a small sample, usually a rectal biopsy, we culture, a week later we can do the assay, and it costs about €1,000 compared to if you would give the drug to a patient, it would cost, well, now the prices in Holland are lower, but in the US it would cost $500,000 to know if it works or not. We started this with this guy. So Fabian is a patient who has a unique mutation shared only with one family member, an aunt. The aunt is clearly 30 years older, does quite well. He was very, very sick. His doctor realized that the mutation in this family mapped close to the mutation that was responsive and was tested in a trial by Vertex. Called him up, and we tested him, and he responded fantastically well to this single drug treatment. He was then put on the drug, and essentially in a day he had his breath back, as he said, and he's now back on the soccer field. De Wereld Draait Door is the most famous Dutch talk show that only a few in the audience here will ever have seen. But he told his story, and based on this, the Minister of Health then said in Holland, okay, every time that in Utrecht these people find a positive organoid test, insurance companies will have to pay for the drug. So far, out of the 1,500 patients, we've probably tested about 200 or 300. 60 of them were in the rare category and responded. There's also about 2/3 of the rare category patients do not respond to this particular drug. We know there are additional drugs being developed. We have already tested them, and we have more patients that will respond. Now, you have to realize these patients in the Western system will never have access to these very expensive drugs because you cannot do a trial on one guy. And we're still now legally, so the insurance companies now want us to screen the large group, about 600-700 patients, where we know statistically only 200 will respond, but they all will want this drug because there's nothing else, very little side effects. And there's now a sort of a legal interaction with the Cystic Fibrosis Patient Organization and the ministry and the insurance companies to see, and actually it was a big fight with Vertex over the price for this drug, and now the deal is that we will screen. All patients will get the drug, but if our test is negative and clinical results are unclear, that the drug will be stopped. And then these patients will be in our biobank, and we will test them against additional drugs. So this looks like it's really working well. And then very early on, this is 2013, I had never heard of CRISPR-Cas9 at that time, but one of my postdocs, Bong-Kyung Ko, had somehow laid his hands on the kit. I don't know how he got it. And then he showed me this experiment where he had caused a double-strand break in, I believe, Fabian cells, but also in some others. And very simply, these organoids are extremely good at homologous recombination. He could sort of bridge the gap with a small piece of DNA and then replace the mutant sequence by the wild type sequence. And in his first try, he did 3 or 4 patients. In all cases, did he get multiple clones of organoids in which he had repaired the original sequence. And this is the outcome. Of the test. On the left you see, I believe this is Fabian's rectal organoids again, and on the right, after we've gone through this CRISPR mediated gene repair strategy, works very well. So the next thing would be that we give this back to Fabian. Obviously there's going to be huge regulatory hurdles, but also this disease is not only a lung disease, they have troubles in the liver, in the pancreas, in the gut. So we're now pursuing this for simpler genetic diseases that affect the liver, where we know we can grow these organoids very well. There's a simple disease, and we also know when we get a few normal stem cells back into the liver, internally they will expand and they'll replace all of the affected stem cells. Some other applications. This was another dogma that cancer comes about by mutations, and that cells that proliferate rapidly would accumulate more mutations than cells that don't proliferate. I guess many of you believe that this is true. Now, it's impossible at the moment to take a single cell from your body and determine the mutational load in that cell, because the technology don't exist to do a whole genome sequence with high enough quality to call mutations in a single cell. And what we decided to do is actually to take single cells from individuals of various ages and then briefly expand them in culture as a clonal organoid, assuming that the mutations in culture would be, the numbers would be so low compared to what you would see in vivo, that we would get a snapshot of the state of that single cell in vivo. And that's actually what we found. And we started with 3 tissues, colon, small intestine, and liver. Liver would be a non-dividing tissue, so the assumption would be we find very few mutations here. Small intestine and colon, we think both have rapidly proliferating stem cells, but Bert Vogelstein had this paper stating that the stem cells in the colon were extremely active and the ones in the small intestine were not, which surprised us, but that would explain why we get colon cancer and we don't get small intestinal cancer. For the cancer people here, this is the Tomasetti paper in Science that has led to lots of discussions. So we took a number of stem cells from these 3 different organs, straight out of patients, expanded them clonally, sequenced them. The individuals were of various ages. In the middle here you see small intestine, babies, old people, everything in between. And what we find is essentially that as you sit here, every week of your life, you add a single base change in your genome, in your whole genome of each of your stem cells in the gut. So this is about 40 to 50 per year, and you see that here. Small intestine and colon don't differ, so the rate of mutation is exactly the same. The rate of mutation in this case does not explain why we get colon cancer, we don't get small intestinal cancer. There must be microbiome effects, or there must be chronic inflammation that allows the mutant cells to grow out. And the liver, we now have more data points, is exactly the same straight line. So independent of whether your cells divide or don't divide, every cell will sort of incur a single base change almost every week of your life. This is, of course, comparing a little bit apples and oranges, but at least it underscores that a strategy to not divide, which many stem cells biologists believe in, doesn't really help, because if you don't divide, you don't clean your genome as well as you do when you do divide, and it doesn't protect against mutations. So that's the normal rate of mutations. We're now trying to see this in other organs to see if it's always the same steady rate of 40 mutations per year. We've also, this is a paper we're trying to write up with Mike Stratton. We did something very similar for 3 primary colon cancers. We divided primary colon cancers up in several sectors, and then from every sector we picked single stem cells. At the same time, we picked single stem cells elsewhere in the gut of these patients, so we would have a snapshot of the normal stem cells and the cancers that's presumably derived from this normal stem cell. We can sequence them. Also, on top of that, we do have the cells alive. It's not just a DNA sequence, but we have them cloned, we have their sequence, and we can now expose them to drugs, which actually gives us a very sobering message. But the first thing is that, and this has been seen by others, when you take small bits of a tumor, you get a less defined mutational spectrum than you would get from single cells, but the message is the same. There is a dramatic diversity, intratumor diversity of mutational states of single cells. Now, this is one of these tumors. You see 4 normal cells. The length of the bar is the number of mutations. The color of the bar is the type of mutation. Mike Stratton has come up with this system where he defines signatures. So C to T is one signature, but then the base in front and the base behind will give you 64 combinations of triplets. And then you can look at the middle base and see what has changed in. This defines mutational signatures, and these appear to represent mutational processes like smokers or UV radiation or MLH1 deficiency, etc., etc. Now what you see is that this orange is a methylated C that decays to T, is the normal mutational process, but somewhere in this tree, and we can almost calculate the age of the patient when this happened, something happened, and this was the mutation of 3 or 4 genes in the same cell. So presumably there's a clone of cells, second mutation, again a subclone, third mutation, a sub-subclone, and eventually you get an explosively growing tumor. And interestingly, 80% of the, for instance, in this particular clone and the other clones from this region are similar, yet not identical. 80% of the mutations are unique to this branch and are not shared with the other branches in this tumor. And you'd see additional oncogenic mutations in branches of the tumor. Now these are 2 other patients, same thing, there was a whole genome duplication here. So this shows actually that also an assumption, but never formally proven, that cancers are more mutagenic than normal cells. Because the normal cells, so these cells are the exact same age as these cells are, but you see the moment they became a cancer cell, they activated many additional mutational processes. Some of them we know the causes, others we don't know the causes, leading to a 10 to 100-fold increase in mutational activity, and then an enormous diversity in mutational profiles per cell. What I don't show here, also when you now take these clones and you expose them to all of the available colon cancer drugs, and these patients had never seen chemotherapy before their operation, you'll find clones that are entirely resistant to any drug that you would give to this patient. Very, very sobering, because and it's not always the same clone, so you'll find clone 3 and 10 will be resistant against 5-FU, clone 7 will be resistant against cisplatin. You could come up with, potentially with combinations of chemotherapeutics that would kill all the clones. I have to stop almost. I'll do one more experiment. But the patients would never tolerate that. Well, finally, we can model cancer now in the lab. We can take, so as you know, it's a mutational process involving 3 or 4 mutations. What we did is we took the 4 most common mutations in colon cancer, realizing that they actually exactly match the growth factors in our, which we totally empirically defined, the growth factors in our cocktail. So APC is a negative regulator of Wnt, KRAS is in the EGF pathway, SMAD4 is in the BMP pathway, and essentially when we target these 4 genes, we can step by step take the growth factors out of the medium, and then this is for instance Wnt plus Wnt, with Wnt they die, human colon organoids, when you hit APC, they will grow out clonally. Next, KRAS, without EGF, normally they die, but if you hit KRAS by homologous recombination, they will now grow without EGF, they only need noggin. P53 we can kill with nutlin, so normally nutlin kills them, but now we can make P53 mutant clones. And then finally SMAD4, which would be the final nail in the coffin of this cell. Normally without noggin they die, but if you take out the transcription factor in the BMP pathway, SMAD4, they will happily live. And only with the combination of 4, so they're now fully independent of any growth factor. We can just, they need extracellular matrix, that's all. But when we now transplant these cells anywhere in mouse, they make a fully invasive and metastasizing tumor. And any combination of 1, 2, or 3 mutations can only grow orthotopically, will not invade, and will definitely not metastasize. Now this one I cannot show. We built large biobanks. Now we have hundreds of colon cancer cases, hundreds of breast cancer cases. The paper will come out in Cell. Ovary is coming up. We have lung cancers. We have pancreas cancer. We have prostate cancers. And essentially we think we can grow every tumor from any of these patients, and then very similar to what we've done with CF, we now hope in trials, we hope to match them to the best chemotherapeutic for individual patients. In the backs of our minds, we realize the heterogeneity in this tumor, so there will always be a complication. And eventually we hope this to find better rules as to what chemotherapeutic should be given to what individual colon cancer patient. With that, I just name Toshisato. Well, first Nick Barker, who spent 10 years to find LGR5, following many false leads, and he's now independent in Singapore. And Toshisato, who, against the advice of all his colleagues in the lab, decided to see if he could grow these colon stem cells and come up with this organoid technology. Thank you very much for your attention.